Hello, everyone. Welcome to Foresight's Biotech Group. Today, we have Chiara Herzog on with us. She is a King's Prize Fellow at King's College, London, and she's the inventor of multiple patents that leverage the epigenome for disease risk prediction. And also, importantly, she's a 2025 Foresight Biotech Fellow. So without further ado, Chiara, we're so excited to hear what the epigenome remembers.
Thank you so much, Lydia. Okay, let me share my slides. And as a disclaimer, so I've already mentioned this to Lydia, but I have been up since 3 a.m. my time and it's currently 7 p.m. So I've had a lot of coffee. If I don't make sense, that's probably why, but hopefully it still makes sense. So as Lydia already mentioned, I'm currently based at King's College London, where I'm starting my research lab. And a lot of the work I'm presenting today was done at the University of Innsbruck. So there's a little bit of overlap in those institutions. And I'd like to start my talk with one of the very fundamental kind of mysteries in biology, which is how the same genetic code can give rise to such incredible diversity in cells. We have, I think, over 200 cell types in the human body that are sharing the same genome largely. I'm mostly interested in the basis of human aging and disease, and we know that genetics does play a big role for this, but so do diet, environment, and lifestyle. And what's really exciting is that actually a lot of these, or all of these, come together and are integrated on the epigenome. We know there are several epigenetic mechanisms, and just for those of you who may be less familiar with them, there's really almost an order of magnitude difference. So even from basically how the chromatin is organized down to how the DNA is wrapped around its nucleosomes, as well as modifications on the DNA sequence itself. And this is really what epigenetics means on top of the genome. So we can think of it as essentially the software that's programming all of our cells on top of the hardware, which would be the genome. That's the kind of hard-coded instructions. And a lot of my work is really focusing on DNA methylation specifically, which is one of the best characterized epigenetic modifications. And I just wanted to mention that this is a very tightly regulated process. So we have enzymes that are controlling this process in all of our cells that can either add methylation. Sorry, let me actually make a laser pointer so you can see where I'm pointing at. So we have enzymes that can methylate de novo, so that adding new methylation, or maintain, or also demethylate. And I wanted to mention that active demethylation is actually a multi-step process that also involves base excision repair, which is essentially kind of an expensive process that cells don't really want to do unless they have to. So what that is to say is that epigenetics really does encode lasting memories that our cells want to retain. So coming back to this integration on the epigenome, my work and also that of others has shown that the epigenome can tell us about past, present, and future. What do I mean by that? It can tell us about past exposures, including smoking, viral infection, hormone exposure, and even early life adversity. It can determine present cellular state. So we know that DNA methylation and also other epigenetic modifications influence gene expression, and they may restrict future cell plasticity. And we can also leverage this to predict future disease risk. So a lot of my work has focused on women's cancers in the past, but we also know that we can predict mortality, aging, and age-related disease. If we think about the very first slide that I showed, which was all of the different cell types, I wanted to highlight one thing, and that is a lot of the studies that historically have looked at DNA methylation have looked at epigenome-wide association studies done from blood samples. And that makes sense because those are quite readily available. However, if we think about all the different cell types that are in the body, they actually have quite distinct epigenomes. So there may be a way that actually all of these exposures that we have interact with our cells in different ways and leave distinct patterns. And that is what a lot of my work has explored. So let me move on. So for instance, in the context of smoking, this is a paper that we published last year. We looked at how smoking influences different cell types across the body. So looking at both those that are essentially directly exposed to cigarette smoke and those that may not be exposed. And we use computational deconvolution algorithms to see how different lineages of cell types would be affected. And what we saw here is it was actually quite interesting because the same sites may be affected differently depending on the tissue you look at. So here, each column is a tissue type and each row is one exemplary CpG methylation locus. Using DNA methylation, it's nice because you can actually, for instance, tell how large your immune cell proportion is in a sample. And we found that some sites are essentially changing methylation only in epithelial lineage cells, but not in blood. So for instance, this first locus, we can see that with decreasing immune proportion, we see a bigger difference in methylation between never smokers and smokers. And then there was also some where it was actually the opposite. So for instance, for the second row, we saw that consistently with increasing immune proportion, we saw a bigger difference. So we wanted to look at this in a slightly more systematic way, and we saw whether we could cluster these groups of CpGs. And this gave quite an interesting picture because we saw that there were some sites that were essentially losing methylation with epithelial cells, regardless of where they were in the body. And these were generally associated with detoxification response genes, which was a very nice sanity check because it makes sense that when you smoke, there's some sort of detoxification going on. And actually, with immune cells, there was a loss of methylation in different loci, and those were primarily located in developmental related genes. And then there was one group of methylation loci that was specifically gaining methylation in cells that are lining the oral cavity, so oral epithelial cells. And they really gained methylation in loci that were related to cancer pathways. So that was obviously interesting because we saw very distinct impacts of methylation changes in different cell types. We then had a collaboration with a group in Bristol who collected a data set from people who never smoked, current e-cigarette users who smoked fewer than 100 cigarettes in their lifetime. So they were essentially non-smokers that started vaping, and current smokers. And when we looked at the immune-related changes, we saw that, as expected, the smokers lost methylation at these sites, but there was no difference between never smokers and e-cigarette users. However, when we looked at the cancer-related pathways that are lining the kind of in the cells that are lining the oral epithelium, we saw that they were gaining methylation in both e-cigarette users and smokers. And that was kind of one of the first times we really found something related to epigenetics and vaping, which had been lost because people hadn't looked at the different cell lineages before. This signature of gaining methylation in cancer-related pathways also predicted relatively well whether a kind of lung carcinoma in situ lesion would progress to cancer or not, which we think could be a sign that it might be associated with a more favorable epigenome promoting cancer progression. However, obviously that needs to be further validated. In other work, we also looked at how different factors kind of influence the epigenome. And we see this almost like these different factors that are kind of shaping and pulling our epigenome. So on one side, we obviously have the aging process, but we also have cell type and lineage, and maybe the diverse functions that a cell is doing or executing. So what we did is we essentially identified groups of genetic loci that are changing methylation with age, those that are associated with different lineages, and looking at different functions. And we selected 3 kind of modules that we were specifically interested in, including stem cell fate and this polycomb promoter methylation. People that are working in epigenetics may be familiar with this. So those are really very tightly controlled, but they change a lot with age, as well as proliferation and senescence. So all of these that are kind of programs that are happening with aging and cancer as well. And some of the findings of this paper, so again, we published this earlier this year, or I kind of wanted to show the highlights because it's quite an extensive paper. But essentially, we do find that lineage-specific age-related changes exist. And of course, there are essentially some methylation loci that are consistently changing with age. And this has been very well described in the literature. But what was interesting is that there are actually some that are changing exclusively in, for instance, epithelial lineage or only in immune lineage. And I think that's quite an interesting way of looking at it because it might be that there are different mechanisms or pathways that are being switched on in these cell types that we could study further. Another thing that was interesting is that actually not all age-related sites are rejuvenated upon reprogramming. So we know that the Yamanaka factors can reprogram cells. However, what we see usually with epigenetic age is that it very drastically decreases over the course of such a reprogramming experiment. And when we look at these different functional modules, we see this for most of them, so proliferation, senescence. However, quite interestingly, with these stem cell fate loci, we saw kind of a transient spike in methylation, which we didn't really expect. And I think that could have something to do with the switch in fate that's happening during this reprogramming. So in this paper, we then looked at a lot of these modules in different conditions. So I don't want to subject you to this monster of a heat map, but I wanted to share one of the key findings that we had, and that was in the context of cancer. So essentially each box here is one different clock. It doesn't really matter so much at the moment what these are, but anything that's red is accelerated aging, anything that's blue is decelerated, and the stars indicate kind of significant differences. So what we saw is that when we look at breast tissue from cancer patients, they showed accelerated biological age compared to control tissue. And this was very expected. It has been demonstrated that cancer essentially looks epigenetically older. But what we didn't expect is that some tissues from cancer patients actually looked younger compared to control tissues. And this was especially the case for cervical samples from breast cancer patients. We essentially didn't really believe this finding when we first had it because, the literature typically says if you have cancer or you have an age-related disease, you're aging faster, and you can measure this in essentially any tissue. But we were fortunate to validate this in a mouse model of breast cancer. So what we did is we were able to induce breast cancer using a carcinogen. And we also explored how a preventive drug would modulate this. So what we saw here, and this is again just a very quick snapshot, is that when we look at the breast tissue where we're inducing the cancer, we again see that this age signature is increased in animals with cancer. But again, in the cervical tissue from the same animals, we saw that the age was reduced. And this was quite striking because it mirrored exactly what we had seen also in humans. It was interesting also with the exposure to the preventive measure because not all of the animals with the preventive measure essentially didn't develop cancer. So those that were exposed but still developed cancer had some changes in their epigenetic signatures, but it wasn't quite as pronounced. However, in those animals that didn't develop cancer, we could see a lot of the signatures that we found were normalized. And with the preventive measure only, there was not as much change. So this is quite exciting because it essentially means that maybe not only can we measure cancer risk at a different site in the body using epigenetics, but we could also track how well preventive measures work. And I think this is quite important also in the context of aging and prevention, where we obviously wouldn't want to do biopsies in the specific tissues. And one of the things that I wanted to mention here is that often we imagine that all of our organs are essentially aging at the same rate, and we can measure this with a single blood sample or a single sample. But actually what's happening is maybe that our organs are aging in a slightly discordant way, and this could harness or encode some information about the disease processes we may encounter throughout life. Very briefly, prompted by the finding that we can detect cancer in different tissues, we did some systematic analysis of which sample types may be most informative for breast cancer detection. So comparing cheek swabs, blood samples, and cervical samples. And one thing that was quite striking, a bit of a complicated diagram, but essentially cervical samples and blood samples shared more information with variable sites in the breast tissue than blood. And this was consistently regardless of which covariates we essentially adjusted for. So we've done a comparison of predicting and detecting breast cancer using these three sample types. And as expected, we found that with blood sample, we essentially get an area under the curve of 0.51, which for those of you that may be familiar with it is essentially by chance. With cervical samples, we get an AUC of 0.66, so around 66% accurate. And with oral samples, this is further increased. We're using a comparative sample size. So this is quite interesting because it essentially means where you're measuring really matters, and maybe the cell types that are in your sample are actually giving you more information for certain disease processes. We've done a lot more on different women's cancers detection and prediction algorithms. So I'm happy to share my slides with you later. We've published a lot on breast, ovarian, cervical, and endometrial cancer. So very happy to go into this in some of the questions later. However, one of the kind of highlights I wanted to share here is for endometrial cancer detection, which is really a disease that emerges with older age in women, and it's also increasing rapidly in incidence because it's linked to obesity. However, there's currently no screening methods, and the way it's detected is essentially taking an ultrasound, which is not very specific, and a lot of women that don't have cancer get flagged and need to undergo biopsies, which obviously is not a very pleasant experience. So we set out that maybe there should be a better way to do this with all the information we have now. And what we found is that when we look at cervical sample methylation, there are a lot of regions that really show quite high gains in methylation in individuals with endometrial cancer. So we basically developed this into a PCR-based test because we wanted to make this quite widely available, and the differences are quite striking. So it's relatively easy to do. And we did some initial validation in a paper that was published in 2022, where we compared our test with ultrasound as well as a different new tests based on DNA mutations. And this showed that our tests outperformed the other two methods. And also in a biobank, it seemed to predict cancer before it was diagnosed clinically. So that was quite exciting because it means we could pick it up a bit earlier than that. And we since then followed this up at a larger study conducted at University College Hospital in London. And this is just kind of a outtake from the figures that we had there. But this is essentially a map of the false and true positives with the respective diagnosis methods. So in the same women, they were recruited in a clinical diagnosis pathway. So this is all real life data. And we asked the participants to essentially take one swab sample before their entire pathway. On the top panel, we have the essentially out of 100 women, how many would have been false positive with ultrasound are these women that are in kind of the red outlines. So we have 53% false positives, whereas if we use a methylation, we actually only had 2.6%, which is really a huge impact if you think of reducing biopsies and invasive procedures in the future. So this test has actually now received approval in the UK for the Medicines and Health Regulation Authority, and we're implementing this further. So I just wanted to take a step back and think a little bit about aging and prevention and how we want to move forwards. And I was like showing this image because it's something that we all know. It's a blood pressure monitor. And in cardiovascular disease, we know that elevated blood pressure is a risk factor, but we can measure it and we can do something about it and we can quantify whether that worked. But again, in aging and even in cancer, we're not really there yet because it's quite hard to really say how fast are we aging, and on the other hand, what can we do? So we know that, again, health lifespan has increased quite drastically over the last 100 years, maybe, but that hasn't always translated to extended health span. I think I'll go over these quite quickly because I think the crowd here is very well familiar compared to some other crowds. And the lifespan in Europe, but also in the States, obviously is never a match for the actual years of healthy life that we have. So we usually spend around 20, 30 years with at least one age-related disease, and we really want to avoid that. And the other thing we really want to avoid is extending this period of life. So we don't want to spend more of our years with disease. And this is unfortunately currently where medicine is going because we're really treating symptoms after they appear. So one thing that we would like to do is extend this healthy lifespan as much as possible. And one thing that I'm very passionate about is, for that reason, of course, geroscience and the geroscience hypothesis to treat the underlying processes that power all of these age-related diseases. I think one thing that is important is obviously reversal is extremely powerful. But on a global scale, I think prevention is still a very powerful approach as well, because if we treat before the onset of age-related diseases, I think that's going to make a big impact for population health. I think these interventions may be different depending on the personal background, including maybe genetics, maybe the life stage that you're in. So I think one thing that we really need is to have some tools that can measure aging. So this was a very roundabout way of saying we need biological age measures. And again, most of you will be very well familiar with this, but we have now great tools, including epigenetic age clocks that correlate very well with chronological age and can tell us about different disease processes. We know there are now different generation of clocks depending on how they were trained. I think there have been a couple of seminars on this topic already, but I just wanted to mention that obviously some of the earlier clocks were trained on chronological age data, whereas some of the newer clocks are trained maybe on time to event data or even the rate of aging. So there's a lot of different clocks that we can measure. It's not always clear which ones, and I'll come back to that later. And at the moment, what we're often doing is that we're essentially saying, okay, you have a lower than expected epigenetic age, you're aging slower, or you're maybe aging faster. But I don't know if that's really the best way to go about it. And even showing some of the data that I had before from the discordant aging study where we looked at the breast cancer, that is maybe throwing up some questions if that's the right interpretation. And as part of the Biomarkers of Aging Consortium that I'll come back to in a second, we had a paper last year where we looked at the biggest challenges that are currently underlying biomarkers of aging to make it to the clinic. Because as mentioned, there are a lot of great research tools, but they aren't necessarily always being used yet. And one of the key challenges we identified was essentially that we have a biomarker level, but at the moment it's not always clear what to do. So let's say you have an accelerated epigenetic age. What do you do? Do you maybe eat healthier or exercise? But you probably already know that you should do that anyway. So what we really would like to do is have a clear intervention or actionable insight. So some of the questions that I've also been quite interested in answering is how can we interpret these epigenetic clocks and which other features are they associated with, and also how malleable are they? So can we really change our epigenetic age over time? And to answer this question, at the start of my postdoc a few years ago at the University of Innsbruck, we initiated a new intervention study where we tried to answer this in a very systematic way. This was supported by the Human Exposome Assessment Platform and the European Research Council. And we essentially recruited healthy women. So our focus was still very much on women's health and women's cancer prevention. And we essentially asked them to either do intermittent fasting or smoking cessation based on existing risk factors that they had. We collected essentially any sample that we could non-invasively, so saliva, buccal swabs, blood, cervical swabs, urine, fecal samples, clinical data, and even skin biopsies. And we collected those longitudinally, so every 2 months over 6 months. And we subjected them to multiple omics. So sorry, I think this is quite small on the screen. Oh God, okay, I've done something stupid. So those are quite small, but we essentially did DNA methylation in 3 tissues, metabolomics into microbiome profiling into, as well as very detailed clinical profiling. And together with the interventions that we did, we think we have essentially almost this like 4-dimensional data set that's encompassing time, different omics, different samples, and different interventions. We collected over 3,400 samples, and what that now allows us to do is explore how malleable some of these biomarkers are. So can they really be shifted? Of course, what do these health interventions do? And also, how do changes in epigenetic biomarkers maybe correlate with changes in other systems? So let's say immune cells or maybe microbiome, etc. So some of the things that we can do at a very basic level is just look, okay, how variable are these different features overall? And essentially, we can then rank them. So what we saw is that the saliva metabolome and also the urine metabolome were essentially just very variable even within an individual over time. And that makes sense because actually when we looked at the features that we have in them, they're very driven by diet and recent intake. So depending on maybe what the people were eating the day before the visit, that would change. And then on the other hand, there were some features that were a lot more stable over time. And here we had actually blood methylation biomarkers and also some of the immune cell markers, which again makes sense because typically your immune populations that you have in the blood aren't shifting a huge amount from one visit to the next, which probably would be quite unhealthy. One thing that I wanted to mention is that actually in the smoking cessation arm, we were able to see whether the features that I introduced at the very start of my talk were changing over the course of the intervention. And we did indeed see that. So for instance, looking at this epithelial detoxification signature, we saw that in individuals that had lower compliance, so essentially they didn't give up smoking, we saw no significant changes, whereas in individuals that did give up smoking, even after 2 months, they already showed a reduction in this signature. And it was a bit more variable depending on the type of signature that you looked at. But essentially, that's in line with also some of the literature that giving up smoking does reverse some of the changes that we observe. We were also able to look at, for instance, how changes that we see over the 6 months would relate to the expected change that we see with aging. So this is quite a complicated figure. Again, very happy to share my slides, or you can scan the QR code because actually these are all available as preprints, so you can also read them. But we saw that a lot of the features that we measured, especially with intermittent fasting, were changing in an age-opposing or attenuated manner compared to what we would expect. And this was actually increasing with more compliance. So that was quite a good sign, saying essentially intermittent fasting does seem to attenuate some of the age-related features that we have. But then one thing that we looked at was also detailed immune populations. So I think in total, we measured around 50 or 60 different populations using a very detailed flow cytometry panel. And as you can imagine, this is quite a lot of data. So we're like, okay, how can we maybe make this a bit simpler? And what we did is essentially generate this immune age metric that we derive at baseline, so we can predict actually from different immune cell populations how old someone is. It doesn't quite work as well as epigenetic clocks as expected, but you can still, it still works pretty well. And what was interesting is that actually over the course of intermittent fasting, the immune age seemed to slightly increase, if anything. So this was a bit different to a lot of the other features that we saw. So most of the microbiome and metabolic features were shifting in a kind of beneficial direction, but the immune system seemed to do kind of its own thing. So that was interesting. We haven't followed this up further, but one thing we wanted to show is that actually this immune age metric that we defined, although it was only encompassing different cell populations, it also correlated with how much essentially pro-inflammatory cytokines these cells would release after stimulation. So it did seem to encompass some kind of inflammatory aging component. Great. We can also do things like multi-omic integration. And for the interest of time, I probably won't go into too much detail, but there's a lot of data that we're really following up here on this side. And one thing that I'm quite interested in, and this is one of the really great selling points of this study, is that we have all of this data from the same people over time. So we can then see changes in which biomarkers change with each other. And we can build these networks to essentially check, okay, which changes are happening together. So here we have some of the biomarkers that we all know, and we see, for instance, grim age and Dunedin pace, and also pheno age really seem to associate quite well with changes in immune cell proportions, which is essentially as expected. To look at this in a more systematic manner, we can explore across a range of different biomarkers how many significant associations do they have, and in kind of what dimension are they. So we see that for blood biomarkers, they generally do seem to have more associations, but actually a lot of them are in like the blood hemogram or blood tests. So for instance, one thing that was quite a nice finding is that I think these are predicted neutrophil proportion based on DNA methylation data. And this is the true neutrophil count, and we can see they correlate really well. So this is nice because actually a lot of our assumptions are relying on this. So that was great. But this also showed that some other aging biomarkers are quite strongly dependent on these cell type counts, which again is known, but it's a very nice sanity check. And we can then start exploring, for instance, how does grim age accelerate with maybe microbial features that may participate in the aging process. So this is a very comprehensive study. I've not really done it justice by skipping over it very quickly, so I apologize for this. However, I've mentioned the preprints are available. And we also want to share this resource with the community, although we can't deposit everything publicly. But we have made a lifestyle atlas where people can explore essentially how features are changing over the course of interventions, and very happy to talk about this afterwards. Great. So finally, I just wanted to talk a little bit about some of the community initiatives that I've been involved in, and hopefully many of you already know about the Biomarkers of Aging Consortium. But those of you who don't, so I've already mentioned there's a ton of biomarkers of aging, and that's a good thing, but it's also not always great because it makes it quite hard to see which biomarker should I use, when do I use it, how can we translate them to the clinic. So we were founded in 2022 with the goal to advance these biomarkers into reliable tools for longevity interventions. So we have a really great steering committee, scientific advisory board, core support team, and obviously executive committee working towards these questions. So some of the things that we do, we're building community, and that's, I think, maybe one of the most important aspects, to bring people together to speak. So we host a conference every year, and this was actually just a few weeks ago at Harvard Medical School. I think we had around 400 attendees, and we'll be back next year. So please do keep in touch if you'd like to attend. We're also promoting open science. So we just recently had a manuscript accepted for our open source Python library called BioLearn. And that makes it very easy for people to compute biomarkers on their own data sets. I think we have over 30 different epigenetic biomarkers, and we've actually worked with developers to check that what our library gives you is correct, because that's not always easy with different processing pipelines. We're also hosting a data science inspired challenge, giving away money to the best biomarkers. We're hosting our own longevity study, and we're also working on consensus with that community. So essentially identifying what are the pain points and how do we overcome them together. We've published a few manuscripts over the years, so I can share the links to them later, but we had manuscripts in Cell, Nature Medicine, Nature Aging. We've also recently worked with the Longevity Biotechnology Association to see how can we standardize sample and data collections in early stage clinical trials to promote interoperability. So essentially saying, okay, we can compete on the targets that we're working on, but maybe we can standardize on the tools. And we're also working with XPRIZE on best practices for immune aging biomarkers for their health span prize, which I think should be quite exciting. So we'll be submitting that very soon. This is a very last minute update, but we had a very exciting initiative that was actually supported by Foresight. So I just wanted to mention this. So I'm based in London, and we now hosted our first London Aging Research Network inaugural symposium 2 days ago, which was a great success. And we actually hosted a funding pitch competition, which was really exciting. And we'll have some new data on omics and ovarian aging. So that should be very fun. Great. And then let's end up. So what are the future directions? I've kind of given a very chaotic overview of a lot of different things that I've done. But one thing that I'm really passionate about is working on epigenetics to unlock the epigenetic code, but maybe moving from correlation to mechanisms, because I think that's one of the most powerful aspects that we could unlock for the next therapeutics to target aging. And thinking back on all that epigenetic does and integrates, it's really a very powerful molecular archive of a lot of different things, including cell lineage, cell fate, exposures, aging. And that raises the question of whether it's maybe not just a quantifiable measure for disease risk, but could also mediate health effects of aging and therefore be a therapeutic and preventive target. With that, I'm at the end of my talk. I'd like to thank all of our mentors and colleagues and collaborators that have enabled this work, and I'm looking forward to your questions.
Thank you so much. I have a small question. This was something that you mentioned at the beginning of the talk, and that you said it's very expensive for a cell to demethylate. I was wondering if you could say a little bit more about what that entails and some details. Yeah.
Yeah, absolutely. So expensive is maybe like a weird way of phrasing it, but essentially it's not something, so it's kind of energetically and metabolically expensive, if that makes sense. So they have to go through this kind of multi-step enzymatic process. And that means it kind of takes a while. It also involves some almost DNA damage response. And cells don't really want to do that because that always entails a risk of mutagenesis. And it's actually interesting because there's also now some research that methylated CpG, so these are the cytosines that are methylated, mutate more frequently. And it's not always clear if that's coming from this demethylation process or that's independent. So there's some link also with more frequent mutations. And yeah, so that's essentially why this isn't done lightly. Obviously, cells can do it. And this is why epigenetics is great, because it's dynamic. So it's possible, but it's not too dynamic. Because if you're thinking of, for instance, gene expression, that can change super quickly. But it's maybe more difficult to measure like the more long-term effects. So this is why DNA methylation is great.
Thank you. Creon, would you like to ask your question?
Sure. Yeah. Great talk. Thanks for everything. I'd like to ask if you have done research, or if anyone has done research that you know of, on using these kinds of analytical techniques to look at two special kinds of cells. One would be the really fast replicating cells, the ones that get, like stomach lining and these kinds of things, and stem cells too. Stem cells in particular, they must have a whole different methylation situation. And then also gametes, because, there's this whole mystery, right, of like how two mature adults can, with their aged methylated genomes, can produce an offspring which has a fresh one. Like, what's going on there? And so have you guys looked into that or anything like that?
No, that's exactly right. And this is actually one of the things that I'm really excited about, because obviously there's a lot of exciting work from Steve Horvath, which has shown this pan-tissue blocks that work across all tissues. And that does seem to show that there are certain sites that are shared across tissues and even across species and different mammalian species. However, I'm really interested maybe in almost the opposite end of that. So how do the different cells, as you're saying, that are maybe replicating fast or not dividing at all, so thinking of gametes or neurons as well, which obviously age, but it's not really clear why, and actually would be quite interesting to dissect that. And that's something that I will be exploring in the future. I'm sure other people have looked at it, but I think a lot of the limitations have been that now we're really at a point where we can scale up these analyses, because we have arrays that are becoming more cost effective. And we also have this insight that, maybe tissues are aging differently. So I think this is something that's going to be very exciting in the next couple of years.
Yeah, well, stem cells also, let's add them to the list, especially if you can go into like a whole tree of different, potencies.
Yes.
Anyway, another thing that is related to this, which I'd like to ask you, is, so can I say this? What is the evolutionary, or not, like, what is this this methylation is a huge topic, right? Because it actually controls, or to some extent controls, differentiation and things like this. So it obviously is essential at some level, but also it's associated with aging and senescence and this sort of thing. So I guess what I'm wondering is, how do you separate that, or do you have any thoughts on that? Like what is adaptive methylation and what is maladaptive methylation?
Yeah, exactly. No, you're raising excellent points, because that's actually one thing that I'm very excited to explore in the future. So I think that essentially my hypothesis is that the stem cells, and initially as we're young, these profiles are very cleanly defined and very neat. And then as we get old, as cells replicate, maybe that signal becomes a bit more noisy and a bit more muddled, which on one hand might mean that maybe the cells are less able to adapt, or this, the kind of differentiation profile is less clear. So they're maybe also doing their function less well, but it's not clear why that is, and how maybe, every baby is born young. How do they manage to really rescue that and start from scratch? I don't know, but it's very exciting.
So another question, and then I'm done, is, and you can answer this privately later if you wish, or publicly now. What do you think of the work of David Sinclair? Because he's certainly kind of all over this stuff.
I think it's very interesting, and I would love it if it were true, because one of my ideas is that epigenetic aging is a driver of aging phenotypes. I think one of the challenges is that obviously the ice mice that were created are induced via mini DNA damage, which they say isn't mounting that response, I don't know. So I don't think I'm familiar enough with the technique. I think there are some potential limitations, but I like the hypothesis.
Well, I mean, he has this claim, which is astonishing if true. And, I don't really, I read this in his book and I haven't read his papers to know to what extent he's actually published evidence. But his claim is that there is some kind of pristine methylation profile that is still carried around somewhere in our cells or in some of our cells, and that to reverse aging, all you need to do is like reapply, like it's not even a theoretical thing. He says it's actually there, and just you have to somehow replace the senescent methylation pattern with this healthy one, which somehow is there because it's put in place in the gametes, for instance.
Yeah. I don't know. So I don't know. I think it's a very intriguing hypothesis. I think nobody knows where this would be stored at this point, but I think it's a very exciting idea and certainly worth pursuing. So
thank you.
Yeah.
Are you coming to the Vision Weekend or anything like that?
Unfortunately not, because I was in Puerto Rico, but I'll be there next year.
Great. Okay, bye.
Awesome.
Yeah, I've got a question that I'd like to ask, and please bear with me because on some level, I think this is a general question that I don't know which direction it would go, but I'm curious as to whether or not the data was collected in these studies that you were demonstrating to us or displaying to us, where vaccination status for COVID was part of the information collected, and therefore you could potentially look at the role of toll-like receptors and humoral immunity versus cellular immunity in the epigenetic patterns that you're looking at to see whether or not, for example, all of these age-related increases in diseases relating to inflammatory diseases and shingles outbreaks and cancer virulence that are now transitioning from anecdotal reports to published studies, whether or not these have an epigenetic basis that can be characterized that could be potentially used therapeutically in the near future.
Yeah, no, that's super exciting. So for long story short, for the data that we collected and the study that we did, we have some information because actually we started our recruitment during COVID. So I think most of our population eventually had either the vaccine or COVID or both. However, I think for a lot of the studies that exist at the moment, the information is quite sparse and it's very difficult to really comprehensively do that longitudinally because it seems like it's not just acute infection, but it's also infection history that matters. One thing that is super exciting, though, is so where I'm based in London, it's Twins UK. So we have this very exciting twin cohort that was established in the 90s. So a lot of the twins we've actually been following since then, and we have 30 years of follow-up every couple of years. And it's quite exciting because they share the genetic background, but there might be differences in their exposures and also their, health trajectories. And within the twins cohort, we also have a very exciting study that's called Welcome Leap Dynamic Resilience, where we're looking at how within twin pairs, twins respond to, for instance, COVID-19, because we know that even within people of the same age and the same genetic background, they might have different responses. So that's something that we're very interested in exploring. And we've currently generated already a lot of multi-omic data, which is ongoing with the analysis. But I think that should yield some quite interesting things, especially because we also have, for some of them, data that precedes long before COVID infections, what happened. So I think that should be quite an interesting integrative model.
A follow-up question. It seems to me awkward to kind of separate all of these different kinds of spike protein influences, for example, on vasculature, inflammatory effects, and potential changes in both acetylation and methylation, sites. But there is an aspect of mRNA vaccination that is absolutely unique, and that is the effect of methyl pseudouridine on any kind of methylation patterning that may be taking place, because there is no methyl pseudouridine effect in COVID or any of the strains of COVID. It's unique to the vaccination process, and that allows you to distinguish between humoral and
Okay, interesting. Yeah, no, that's very interesting. I wasn't aware of that, so definitely should look into that.
Thank you.
Thank you.
Lydia, can I go again?
Of course.
Okay. Another question, channeling my inner Amy Proel here. Another interesting thing, there's no shortage of them, is if you look at her work, and she claims a great deal of what we call senescence has to do with chronic infection or hosting of various parasites, viruses, worms, like the whole gamut of kingdoms. That's another thing that could be looked at. Like, to what extent does infection, disease, and parasite hosting change these methylations?
It's very interesting.
Omics.
Yeah, no, there's actually, so we haven't done a lot of that because we're historically mostly cancer and aging. However, there is one cancer that's driven by infectious disease, and that's cervical cancer. And one thing that we did look at there is essentially is there a signature of HPV infection, and what does that look like in progression to cancer? And I think essentially what we expected is maybe that the signal will get stronger as you progress to cancer. But actually, we saw the opposite. So we saw that in women that were infected with HPV, the cervical cells would show this difference. And then that would essentially get stronger as they progress to pre-neoplastic. But then in women with a cancer, this was lost. So this actually indicates that there was some sort of adaptive mechanism. And essentially, maybe those women who did not mount this response progressed to cancer. That's one example. We haven't really looked at this in further detail, but I think it's very exciting, and there's certainly a lot more to be done, especially as was mentioned with obviously shingles, also Epstein-Barr virus. I think there's like a lot of interesting evidence coming out how those link to chronic disease.
All right. So that puts us just about at time. If there are any other questions, speak now.
Or email me.
Or email Chiara.
Thank you.
All right. Well, thank you guys so much for joining, and wishing you a great rest of your day. And see you.
Thank you so much.
Thank you, Chiara.
Bye.